Imagine trying to put out a fire, like a really roaring fire, inside a locked subterranean bank vault.
Which is, pretty much impossible.
Exactly. I mean, you can spray thousands of gallons of water all over the outside of the building. You can completely flood the lobby.
Yeah.
But the vault is sealed tight.
Right.
And the fire inside remains, like, completely untouched.
Yeah.
And for decades, that has honestly been the frustrating reality of treating complex diseases.
Oh, absolutely. Especially the ones that are hidden deep within the human body or, protected by the brain.
Yeah, we've basically been soaking the outside of the vault. We rely on pills that just manage symptoms. Like, you have high blood pressure, you take a pill. You have a metabolic issue, you take a pill.
It's very much a maintenance mindset. You're just accepting that the underlying fire is still burning.
Which is exactly why you are going to want to stick around for this deep dive.
Mm-hmm.
Because if you want to look at the absolute frontier of medicine, how researchers are actually moving away from that daily maintenance and toward, like, one-time permanent hardware hacks, you are in exactly the right place.
Yeah, it's a completely different philosophy of medicine.
It really is. Today, we are looking at a stack of data covering 3 incredibly cutting-edge phase 1 clinical trials. And what makes this stack so special is that all 3 of these trials scored a massively high 18 out of 20 for their breakthrough potential.
Which is huge. You don't see those numbers often.
Right, but we aren't just going to look at what these drugs are. Our mission today is to look at the evolution of how they are delivered.
Because delivery is really the unsung hero of all medical breakthroughs. I mean, a miracle cure is entirely useless if it can't physically reach the damaged tissue.
So true.
So as we go through these 3 trials, we are going to see this fascinating escalation in targeting. We'll start with a system-wide biological rewrite using just a standard IV drip. Then we'll look at the extreme measures required to bypass the body's natural armor.
And then ending up inside the brain itself, right?
Exactly. Seeing how scientists are actually hijacking the brain's internal neural networks to distribute gene therapies.
Okay, so let's start with that systemic approach. Because the first trial we're looking at is tackling a disease that essentially poisons the entire body.
Right, Fabry disease.
Yeah.
Yeah.
We are looking at the STARE trial, registered as NCT 04046224. It's sponsored by Sangamo Therapeutics, and they achieved a really solid 68 out of 100 overall trial score here.
A very respectable score for an early phase trial.
Definitely. So they are testing a therapy called ST920, and for those who aren't familiar, Fabry is a rare lysosomal storage disorder.
Which sounds complicated, but the mechanics are actually pretty straightforward.
Yeah, the way I usually visualize this is to imagine your body as this massive, sprawling house, and the lysosomes are the garbage disposal units in the cells of that house. Their entire job is just to break down waste.
Right. To put a finer biological point on it, in Fabry disease, a genetic mutation means the body basically fails to produce a very specific enzyme. It's called alpha-galactosidase A.
Okay.
And without that enzyme, a particular type of fatty substance, a lipid, just cannot be broken down.
So the garbage disposals are jammed. And because you can't break down that fat, the trash never gets taken out. Harmful, toxic garbage just starts piling up in every single room of the house.
Which, in a human being, means these toxic lipids crystallize inside the cells of your kidneys, your heart, your nervous system. It's everywhere.
And over time, that buildup causes agonizing pain, right? And eventually profound, life-threatening organ failure.
It does. And historically, the treatment meant you'd have to go to a clinic every couple of weeks for enzyme replacement therapy.
Just continuously getting your system flushed out.
Exactly. You get a temporary shipment of enzymes, they clear out some of the trash, they wear out, and then you have to go right back to the clinic.
A textbook example of that maintenance mindset we were talking about.
Totally. But Sangamo's ST920 is an AAV26 gene therapy. This is where we cross from maintenance into actual biological rewriting.
Okay, break that down for me.
Well, they aren't just giving the patient a temporary shipment of enzymes. They are giving the patient's body the permanent blueprints to manufacture the enzyme itself.
But wait, this is delivered via a single intravenous dose, like literally a standard IV drip.
Yeah, just an IV.
So if the whole body is the house and the toxic fat is piling up in the kidneys, the heart, the skin, literally everywhere, how does one single IV drip of ST920 managed to fix the garbage disposal system in every single room?
Well, it really comes down to understanding the vehicle they are using. So AAV stands for adeno-associated virus.
Okay.
In the lab, scientists take a naturally occurring virus and essentially hollow it out. They remove all the viral DNA that causes disease and makes it replicate.
So it's just an empty shell.
Exactly. And they replace that empty space with a synthetic human gene, the exact instructions for making that missing enzyme,
Oh,
alpha-galactosidase.
hold on. You are saying they are pumping a patient's bloodstream full of
I
viruses.
mean, yes, hollowed out viruses, but yes.
But wouldn't the patient's immune system just recognize it as a viral invader and,
Yeah.
like, obliterate the whole payload before it does anything useful?
That is the multi-million-dollar challenge of gene therapy. And it's exactly why they use this specific strain, AAV26.
Oh, because it hides better.
Sort of. First off, adeno-associated viruses naturally provoke a very low immune response compared to other viruses. But more importantly, the protein shell of this specific virus, the capsid, is highly hepatotropic.
Hepatotropic, meaning it targets the liver.
You got it. It naturally homes in on liver cells. So when the IV releases millions of these microscopic delivery trucks into the bloodstream, the blood just acts as a highway. It carries them directly to the liver.
So the liver just acts like a giant sponge for these viral vectors.
It really does. The liver is incredibly vascular. I mean, a massive volume of blood passes through it every single minute. So the liver cells take in these hollowed out viruses, transport them to the nucleus, and unspool those new genetic instructions.
Wow.
And then the liver undergoes a permanent transformation. It essentially becomes an enzyme factory, churning out a continuous 247 supply of the missing enzyme directly back into the bloodstream.
That is absolutely brilliant. So the blood drops off the blueprints at the factory, and then the factory uses that same bloodstream to ship the finished product out to every single cell in the body.
Exactly.
It clears the toxic fat out of the kidneys, the heart, everywhere. The logistics of that are just incredible. And looking at the trial data, this was a completed phase 1, well, phase 12 trial that actually managed to enroll 36 patients across 18 sites globally.
And I have to say, those numbers are staggering for a rare genetic disorder.
I can imagine.
Finding 36 eligible patients who are willing to undergo an experimental gene therapy, that requires a massive, coordinated global infrastructure.
But it also speaks to the desperation and, the hope within the Fabry community.
Absolutely. And that 18 out of 20 score for breakthrough potential is heavily driven by this exact mechanism. It's a one-and-done systemic fix.
Which is amazing. Systemic delivery is so elegant. The blood is the highway, the liver is the factory. Boom, problem solved. And there's a big but here, this only works because the bloodstream actually has access to the target tissue. What happens when the blood itself is barred from entry?
Ah, now you're talking about the brain.
Exactly. The brain has this biological bouncer, the blood-brain barrier. It is a highly selective filter that lines all the blood vessels in the central nervous system.
And it flat out refuses to let large molecules or engineered immune cells or really much of anything pass through.
Right. Which makes sense evolutionarily. I mean, it's a masterpiece for keeping neurotoxins and pathogens out of our brain tissue.
It is. But as you can guess, when you are an oncologist trying to deliver a life-saving therapy to a tumor located inside the central nervous system, that evolutionary masterpiece becomes an absolute nightmare.
I bet.
An IV drip like the one used for Fabry disease is virtually useless here. The medicine will circulate through the entire body, sure, but the moment it hits the blood vessels in the brain, it just bounces right off.
Which brings us to the second trial in our stack, and the incredibly intense measures required to bypass that bouncer.
This one is intense.
Yeah, we are looking at NCT 041-85038, sponsored by Seattle Children's Hospital. They are targeting pediatric central nervous system tumors, specifically these incredibly aggressive solid tumors called DIPG and DMG.
Heartbreaking diseases.
Truly. And their intervention is a stuck-art T-cell therapy called SCRI-RT CARB7H3s.
Right.
And it scored an impressive 74 out of 100 overall. But when I was looking at the setup for this trial, the logistics seemed almost impossible compared to the last one.
How so?
Well, the Fabry trial cast a really wide net, right? 18 sites globally. But this trial is aiming for a whopping 90 patients, and all of them are being treated at a single hospital in Seattle. Why would they bottleneck an experimental trial like that?
It's a good question. Centralizing an entire phase 1 trial to a single site is a massive burden on the patients and their families. But it's a non-negotiable necessity here because of the delivery method.
A loco-regional delivery.
Exactly. We aren't just hanging an IV bag here. This trial involves placing a specialized catheter, it's often called an Ommaya reservoir, directly into the patient's brain ventricles, or even into the cavity left by the tumor itself.
Wait, so they are surgically drilling through the skull to physically drop the therapy right onto the battlefield?
They have to. Diffuse intrinsic pontine gliomas DIPG and diffuse midline gliomas DMG are some of the most devastating pediatric cancers in existence.
And you can't just operate on them.
No, you can't just surgically cut them out because they don't form a neat little lump. They literally weave themselves into the healthy tissue of the brainstem.
Oh, wow. And the brainstem controls like breathing and heart rate.
Exactly. It's like trying to remove sand that has been mixed into grass without damaging a single blade of grass. It's impossible. And to make matters worse, they are notoriously resistant to radiation and standard chemotherapy.
So if you can't cut it out and you can't poison it through the blood because of the barrier, what exactly are they pushing through this brain catheter?
A living cellular army. Yeah. CAR-T stands for Chimeric Antigen Receptor T-cell therapy. The researchers actually extract the patient's own white blood cells.
Okay.
They take them into a highly specialized manufacturing facility and they genetically reprogram them. They outfit these T cells with new synthetic receptors that are designed to hunt down a very specific protein called B7H3.
I noticed that B7H3 target in the source material. Why that specific protein? Like, if these immune cells are basically doing hand-to-hand combat inside the brain, how do they know what to attack without accidentally destroying healthy brain tissue?
That is the genius of the target selection here. B7H3 is an immune checkpoint molecule that is highly overexpressed on the surface of these specific tumor cells.
Not on healthy cells.
Exactly. It is barely detectable on healthy, normal tissue. So when they infuse these engineered SCAR T cells through the catheter directly into the brain cavity, the cells act like heat-seeking missiles.
That is wild.
They just float through the cerebrospinal fluid until their synthetic receptors lock onto those B7H3 proteins on the tumor. And once they bind, the CRT cell releases cytotoxins that basically punch holes in the tumor cell.
Completely destroying it while leaving the healthy brain cells alone.
Right.
Let me just make sure I'm fully grasping the sheer scale of this operation. A patient, and keep in mind, this specific cohort is recruiting patients aged 15 and older, has to have their blood drawn.
Yes.
The cells are flown to a lab, genetically weaponized to hunt a specific protein, multiplied by the millions, and flown back.
Correct.
Meanwhile, the patient undergoes neurosurgery to install a port in their skull. And then doctors physically inject those living, breathing assassin cells directly into the brain fluid to wage war on a tumor that was previously considered untouchable.
That is exactly the workflow. And, it answers your earlier question about why it's happening at one single site in Seattle.
Oh, right. Because of all the moving parts.
Yeah, the neurosurgical expertise, the cell handling, the specialized manufacturing, plus the intensive care required to manage the inflammation in the brain during this kind of immune response. You simply cannot farm that out to a community hospital. It requires an elite, centralized infrastructure.
It really perfectly illustrates the leap from our first trial. We had a systemic IV fix that relied on the blood. Then we hit the blood-brain barrier and had to pivot to a localized surgical catheter drop.
Right.
But our third trial throws a massive wrench into both of these strategies.
It does.
Because what do you do when the target behind the blood-brain barrier isn't a localized clump of tumor cells, but the widespread deterioration of the brain itself?
You're pointing to the fundamental challenge of neurodegenerative diseases right there.
Yeah.
And that brings us to NCT06064890. This is a gene therapy trial sponsored by Aviato Bio, testing an intervention called AVB101.
And this trial is targeting a genetic subtype of frontotemporal dementia, known as FTD-GRN, which is just a brutal disease. It causes a progressive, relentless loss of neurons across the entire frontal and temporal lobes of the brain.
It strips away personality, it alters behavior, destroys language skills.
Yeah, it's terrible. And the thing is, it's not a localized issue. It's a massive, sweeping geographic problem across the entire surface of the brain. But the delivery method they are using here is listed as intrathalamic administration.
Intrathalamic, meaning an injection deep into the thalamus.
Right, and the thalamus is a structure buried deep down in the central core of the brain.
It is.
So if the whole surface of the brain, the cortex, is what's dying, why bury the medicine down in the core? To use an analogy, if the whole surface of the earth is experiencing a drought, an IV drip is like hoping for rain everywhere.
Sure.
A catheter is like watering one specific garden. But this is like burying a single powerful seed miles beneath the Earth's crust and just hoping the roots somehow find their way to the surface. It just seems completely counterintuitive.
It totally seems counterintuitive until you look at how the brain is actually wired.
Okay, lay it on me.
The brain isn't a static bucket of fluid, right? It's an incredibly complex, highly organized network of highways. And the thalamus sits right in the geometric center of the brain. It acts as the master relay station for the cerebral cortex.
So all the signals go through there.
Almost all sensory and motor information that travels to the frontal and temporal lobes passes through the thalamus first.
Ah, so it's like the grand central station of the brain. Millions of tracks radiating outward to every single neighborhood on the surface.
Exactly. So the researchers at Aviato Bio are taking a gene therapy, another AAV vector, kind of like the one used in the Fabry trial, and they are injecting a tiny volume of it directly into the thalamus.
They are literally hijacking the brain's internal subway system.
They are. The neurons in the thalamus absorb the viral vector, but instead of just keeping the genetic instructions locally, those thalamic neurons read the blueprint and begin manufacturing a protein called progranulin.
The granulin. That's what people with the FTD-GRN mutation are missing, right?
Correct.
So what does that protein actually do for the brain?
Progranulin acts like a survival factor. It's almost like a structural fertilizer for neurons. It regulates lysosomal function. So there are cellular garbage disposals again, and it helps reduce inflammation.
Okay.
Without enough progranulin, the neurons in the frontal lobes just wither and die. But here's the mind-blowing part. Once the thalamus starts manufacturing this progranulin, motor proteins physically carry it down the long axons, the subway tracks, and secrete it out into the frontal and temporal lobes.
Oh, wow. So instead of drilling 100 tiny holes all over the patient's skull to inject the surface of the brain, they just do one deeply targeted injection into the central distribution hub.
Exactly.
The thalamus becomes the factory, and the brain's natural neural networks do all the shipping for you, physically altering the biology of the whole frontal lobe from a walnut-sized area down in the core. That is breathtakingly elegant.
It's an absolute masterclass in biological engineering. And looking at the logistics for this one, they're recruiting 18 patients, but they are spreading them across 19 different sites globally.
Wait, 18 patients across 19 sites.
Wow.
That averages out to less than one patient per hospital.
Yeah, finding these specific FTD-GRN patients must be a massive global scavenger hunt.
It really underscores how rare this specific genetic subtype is.
It does. But what's equally notable here is the timeline. This phase 1 trial actually mandates a 5-year follow-up period for every participant.
Wait, 5 years? Just for a phase 1 safety and dosing trial?
You have to remember the nature of dementia. The degeneration is slow, it's insidious.
Oh, right. It takes time to see if it worked.
Exactly. If you want to prove that a one-time gene therapy injection into the thalamus has permanently halted the physical deterioration of the brain, you cannot measure that in 6 months.
No, of course not.
You need years of careful observation and cognitive testing to prove you have fundamentally altered the trajectory of an otherwise fatal disease. It earned a 73 out of 100 overall score, but again, an 18 out of 20 for breakthrough potential. Because if this works, it entirely rewrites how we treat neurodegeneration.
Okay, so let's synthesize this journey we just took. Because what does this all mean for you listening right now? We started this deep dive talking about the limits of taking a pill every day, just hoping to maintain the status quo.
The maintenance mindset.
Right, but look at the sheer ingenuity of what we just explored. We looked at an IV drip that uses the bloodstream to turn the liver into an enzyme factory for the whole body. Then we looked at a specialized brain catheter dropping living, genetically engineered immune assassins directly onto a pediatric tumor to bypass the blood-brain barrier.
And then the brain's subway system.
Yes. Finally looking at deep brain surgery that hijacks the thalamus, using it as a central distribution hub to halt dementia in its tracks.
It's incredible. And you know what connects all three of these approaches, despite them all being at the earliest stages of human testing, is the pursuit of root cause eradication.
We are no longer satisfied with just managing a disease.
Exactly. Medicine is actively shifting toward permanent hardware upgrades. We are using viruses to write new code into human DNA, and we are deploying living cells as targeted therapies.
It's a whole new world.
It is. Whether it's you, a friend, or a family member, the medicine of the next 20 years will increasingly rely on these highly precise, localized biological tools rather than blunt systemic chemicals.
It really is the difference between continuously bailing water out of a sinking boat and finally diving down to just fix the hole in the hull.
It is. And there's a big caveat here that permanence brings us to a critical, almost philosophical dilemma. And it's something I think is really vital for anyone following these advancements to chew on.
What's that?
Well, If we are entering an era of one-and-done medical procedures where we permanently rewrite the genetic code of your liver, or we install living immune cells that persist in your brain fluid for years, what happens to the concept of a side effect?
Oh, wow. I mean, right. Because if a standard daily pill gives you a rash or elevates your heart rate or, makes you dizzy, you just stop taking the pill.
Exactly. The chemical washes out of your
Mhm.
system, the side effect goes away, you lose the benefit of the drug, obviously, but you stop the harm.
You have an immediate biological undo button.
Right. But consider these therapies we just talked about. If a permanent genetic upgrade to your thalamus has an unexpected consequence a decade from now,
Oh, yeah.
or if an engineered cellular army in your brain decides to attack healthy tissues 7 years down the line, how do you uninstall a cure?
Wow. You can't just stop taking a gene therapy. It isn't a drug floating in your system anymore. It is fundamentally woven into the fabric of who you are.
It is the ultimate double-edged sword of modern medicine. We are gaining this miraculous power to permanently fix the broken systems of the human body, but in doing so, we are completely losing the safety net of simply being able to walk away from the treatment.
That is a fascinating and honestly slightly terrifying thought to leave on. The days of just popping a pill and seeing how you feel are slowly fading into history. We are stepping into an era where we are rewriting the blueprints of the house itself. You just have to make absolutely sure you are going to love the new floor plan, because once the concrete is poured, there is no going back.